Okay, so now we're going to talk about the absorption of our carbohydrates. Now, one thing to keep in mind about absorption of sugars is that not all of the complex carbohydrates that we digest are actually going to be digested within the same rate. Some carbohydrates lead to a near immediate rise in blood glucose, and that's a high glycemic index. Oops. And foods with a high glycemic index are going to be glucose and maltose. And then some with a lower glycemic index, which means they are going to be absorbed slower, are going to be things like yogurt and milk. And so keep in mind that glycemic index is a measurement of how quickly the sugar or the carbohydrate gets into the blood. Now, when we talk about absorption into the blood, there are going to be facilitative glucose transporters, and those are things such as your GLUT1, GLUT2, GLUT3, GLUT4, and you also have your sodium-dependent glucose transporters. And so those sodium-dependent glucose transporters are going to be associated with the brush border only. And so here's our sodium-dependent glucose transporters, and then we also have our facilitated glucose transporters. And you can see that we have our 3 sugars. So remember, we can only absorb monosaccharides, and we have our fructose, we have our glucose, and we have our galactose. So those are the only 3 that we can absorb. We have transporters for them. They are going to be taken up on the brush border side, and then they're going to be released on the serosal side. They can be taken up by either the facilitated or by the sodium cotransport. And the sodium cotransporter is going to only be able to take up glucose and galactose. The facilitated transporter is only going to be taking up glucose and fructose. So what that means is galactose can only enter through the sodium cotransport, and fructose can only enter through the facilitated transport. Glucose, on the other hand, can go both routes. Now, on the other end, they're all going to leave through the facilitated. And so some differences is on the brush border, the facilitated is going to be GLUT5 receptors. And on the serosal side, these are going to be primarily GLUT2 receptors. So galactose cannot use a GLUT5, but it can use a GLUT2. Now, one of the other things to point out here is that you do have to maintain a sodium balance during the fed state because you want to be able to absorb glucose and galactose. And in order to do this, maintenance of your sodium is you have a sodium potassium ATPase. This is going to be on the serosal or the blood side, and it's going to allow the removal of sodium in order to maintain the gradient. So you're going to exchange 3 sodiums out for 2 potassiums in. And this will allow sodium to keep coming in with either a glucose or a galactose. Now our facilitated glucose transporters we've talked about before. There are GLUT1 through GLUT5. One of the big differences is your GLUT4, as we've mentioned before, is going to be insulin dependent. And so GLUT4 is going to be important on your adipose tissue and your muscle tissues. Now, most of the other glucose receptors are going to be constitutive. So GLUT4 is not going to be expressed unless there's insulin. GLUT1 is always going to be expressed because it's on a red blood cell as well as the blood um, in blood-brain barrier and in your brain. And so this is always going to be on. GLUT2 is important because GLUT2 is associated with your liver, kidney, your beta cells, and it's also on that serosal side of your intestinal mucosal surface. And so the GLUT2 is going to have a lower affinity for glucose, which means that it only picks it up when there's a high presence of glucose, the fed state. And then we have GLUT3, which is again going to go back to that neuronal, so that's going to be constitutive. And GLUT5 is going to be on that intestinal epithelial cells, the high fructose receptor, that's what it's called, the fructose transporter. And so sperm also use fructose, and so it's going to be on sperm as well. So again, these are all called different isoforms, and they're going to have different affinities for the sugars. And so just keep that in mind as we go through talking about metabolism. Now, when you get the monosaccharides released into the blood, they then have to be absorbed into the tissues. So these are going to be mostly through your transporters, your facilitated glucose transporters. Liver is going to take this up in GLUT2. Your muscle and adipose tissue are going to use GLUT4, for example, and you're going to have different Km values for these. Now remember that your Km value, the higher the Km, the lower the affinity. So GLUT2 will have a much higher Km than your GLUT1, for example. Now there's also important to just briefly talk about the blood-brain barrier. So there are some differences between your blood-brain barrier because you don't want things to get inside of your brain. You don't want simple diffusion, for example, into that tissue. And so what you're really moving things into is the cerebral spinal fluid, CSF. And then once they're in the CSF, whatever got in can get into the brain. So some of the things about the basement membrane associated with the cerebral spinal fluid is it's going to have tight junctions. And so in your non-neural tissue, they do not have tight junctions, so things can diffuse through, including glucose. The other thing they have is they're going to have a narrow intercellular space, and they're going to lack pinocytosis. Remember, pinocytosis is simply drinking what's on the outside. Your endothelial cells associated with your tight junctions of your blood-brain barrier do not take things up randomly. In other areas of the body, this is done commonly. And so the fourth one is a continuous basement membrane. And in normal tissue, this is discontinuous, again, to allow things to diffuse through pretty easily. What you're going to have to do in neuronal tissue is you're going to have glucose transporters on both sides of the membrane of the endothelial cell. Whereas in this side, the glucose diffuses through, and the glucose that the endothelial cells take up in non-neural tissue, they'll use for energy. So a hypoglycemic response is going to be between 18 to 54 mg per deciliter, and this can lead to a coma. One thing to keep in mind is that your brain is very sensitive to a loss of glucose because it takes longer for glucose to get into the brain through the blood-brain barrier. So that is a barrier. And so your endothelial brain cells are going to, again, have to move glucose from the blood into the brain using the facilitated transporters on both sides of the cell. And then finally, I want to talk just briefly about insulin and GLUT4. We know that insulin is going to trigger that tyrosine kinase receptor, and this is going to stimulate the expression of your GLUT4. So this is just shown here. You have your tyrosine kinase receptor binding to insulin. This is going to trigger a cascade of responses, and one of which is going to mobilize your glucose transporters. Your glucose 4 transporters are on an endosome. The endosome will fuse with the plasma membrane, and then you'll get expression of your glucose transporters, and this will allow your muscle cell to take up glucose. Now remember, the glucose transporter in this case is GLUT4, and this is important for removing glucose after a high carbohydrate meal.